Today, our guest is Tiago Magalhães, and we will be talking about gene sequencing, rare diseases, and much more. Don't go away.
O maior evento para os profissionais da indústria farmacêutica em Portugal é já no dia 6 de junho, em Lisboa. Ricardo Batista Leite e Daniel Guedelha são os keynote speakers de um dia que promete muitas surpresas. Todas as informações e inscrições em www.rethinkingpharma.pt.
Welcome to this episode of the Cruzamento Podcast. Thank you for your amazing feedback. If you enjoy our content, please consider giving us a 5-star rating on your podcast app to support our mission of spreading knowledge. Today, we are exploring the intersection of technology and healthcare. I'm Daniel Guedelha, joined by my co-host, André Correia.
Hello and welcome to another show of Cruzamento Podcast. Today, we have Tiago Magalhães with us. Hello, Tiago. Very welcome to the show.
Hello. Hi, guys. Nice to be here.
Let's start with our very first question in 30 seconds. Who is Tiago?
Oh, Tiago is a Portuguese man. I am 52 and I am a biologist by training, and then I became a bioinformatician. Of course, I am many other things, but I think in 30 seconds, that kind of sums it up.
Beautiful. And let's start to talk about gene sequencing and its a pivotal aspect in our modern healthcare. Tiago, could you share with our audience why gene sequencing is crucial and how it can significantly improve people's health?
And gene sequencing, in a way, is the latest iteration of something that has been around for thousands of years, which is how disease, how our biological body affects diseases. I mean, doctors are asking, what is your family history for many years, many thousands of years, and that's exactly what gene sequencing is, the last version of that. You go into your genes, you understand what your biological hardware is giving out to you. And so in 2000, the gene sequencing the full genome was discovered. And from then on, people could actually go into sequencing their own genes and understand that link to disease or others, like sports ability and so on.
And for those unfamiliar with genomics, Tiago, how could we explain how raw genomic data is transformed into actionable medical insights that can guide patient care and treatment strategies? Because sometimes genomes and DNA is a bit in the air topic. So how does this explain?
Yeah, absolutely. Absolutely, it's not an obvious concept. So the way this works is you take out DNA from your own cells. Every cell has DNA, so you take out DNA, and then you sequence that through sequence technologies that basically decode all your DNA. So that is 3 billion sequence letters, as we say, nucleotides, and at the end you have kind of information, which is basically your genome, consisting of A, T, Cs, Gs, As, Ts, and whatever, and that then is translated through computational, hence the bioinformatics part of this story. So you go from that raw sequence, DNA, machines, raw sequence, and then you need to interpret that, taking out the genes, taking out mutations in those genes, and then people have been compiling for many, many years what those mutations correspond to disease. So if you have a specific type of mutation, for instance, in cystic fibrosis, which is CFT gene, which is an example that people normally use, it's enough to have a few changes in that particular genes to have cystic fibrosis. So people, what they are doing, they go to the CFTR gene, they analyze the mutation, and they say, you are going to have cystic fibrosis. And once you know that, of course, you can start different treatments, you can start on different drugs, you can start on different regimens. And of course, before, people knew that from family history, people had a history of cystic fibrosis, hence you were at risk. But now you go a step further, you sequence DNA, you understand the mutation, and immediately you go through a particular disease treatment.
Yeah, Tiago, interesting, because ultimately what we want is real impact in patients' life and in people's life. One thing that we learned recently as well throughout our conversations is the rare diseases prevalence in the UAE, where you are based. And we'll be very interested if you could share with us what factors contribute to this trend, the high prevalence of rare diseases in the UAE, and how does it drive the urgency of projects, programs like yours, the Emirate Genome Program?
Yeah, I mean, it is known by compiling statistics that the defects in newborns is very high in the UAE, is very high throughout Middle East, Bahrain being the top country in the world with more rare diseases. And that is because for two main reasons. One is there is a consanguineous tradition of the tribes marrying with close families, which then means that genetically mutations can pile up. So that is one reason, and it's well known by, and that's kind of the history and the culture of these countries. And the other one, which is also very interesting, is that there was an explosion in the population of the UAE. The UAE, of course, 50 years ago, the country itself was founded in 71, so 53 years ago, and the population around the 60s was around 60,000 people, and now it's 1 million. So you can imagine that from 60,000 2 generations ago into 1 million, there is an explosion in the population. But the beginning was few people. So one mutation at the beginning, grandmother or grandfather had a mutation, now has many grandkids with the same mutations, right? So that increased the number of rare diseases in the population, and the mutations and the tribal culture of these countries made it a high risk factor for rare diseases. Now, the EGP, or the Emirate Genome Program, was set up to try to use modern technology to sort out those issues, right? And the way you do it is, of course, through genome sequencing. You know those mutations, you know the risk, and you can do 3 things. One is premarital screening, which is you advise couples that are getting married to get sequencing, and then you understand the risk of those mutations to become a child, an offspring with rare diseases. So that's one. And as a historical example, Tay-Sachs disease in New York in the Jewish community was actually eradicated through that mechanism of premarital screening, where couples were advised to not get married or to go into counseling or to find ways to mitigate the disease. So there is already a few examples where this has been widely sorted out. And that's one of the main goals of the Emirate Genome Program.
And so we're talking about the Emirate Genome Program, also known as EGP, as you've mentioned, Tiago. And this is a large-scale program on genome sequencing, as you were also explaining. In comparison to others that are being done around the world, and for example, we are thinking about the UK, where there were some news lately where different genome sequencing programs kind of merged together to become a very large program. How would you compare the EGP with these type of programs around the world, namely in the UK? And what are the key differences, and also what are the similarities between these programs? And also, what are the lessons learned from these programs?
That's a big question there. Let me try to unpack all of that. So, in terms of the idea of genomics now has fallen a bit into what people call the PopGen, which is population genomics, right? Where you go into a country and you sequence a very large proportion of that population, and then you have the variance of the genomes, right? Because this goes back to when you have a population, right? Not everyone is the same. We all know that, right? So not everybody is the same height, not everybody is the same eye color and whatever, but that's still part of the same population. So this PopGen, this population genomics programs, trying to understand the genetic variance or differences and similarities within a population, because you need a very large number of people to do that. And UK Biobank and Decode in Iceland were pioneers in doing that, where they took a large proportion of the population and understood that. The difference for it and all of us, for instance, in the US is another program where they're trying to sequence 1 million people. The difference in this population is the proportion of people sequence. So here we will try to sequence 1 million people out of 1 million population, which is effectively 100% coverage, right? Yeah. And we are already at 700,000 or 650, which means that we are very far advanced into the proportion of the individual in population that have been sequenced. In the UK Biobank is 500,000 people. The new program that you mentioned might go a bit higher, but still the population is around 80 million or 19 million. I hope I'm not very incorrect in the number there. And so the proportion of people sequenced in the population is very different. Now, what that allows EGP to do is to actually be much further into clinical care, right? Because you can have a strategy of sequence once, use multiple times, right? Sequence once, use often. That's kind of the motto. And so if you sequence, that needs in a database every time a patient goes to the clinical care, the doctor can go, get the genetic information and use it, right? In the UK, that would not be the case because it would be 5% of people would have genomes there. So I think that's one of the main differences is the proportion of the sequence. The other kind of technical differences there is that we have long grids, which to a proportion that nobody has. That's kind of technical, but it goes into the sequencing, and having long grids allows you to have more information in terms of structural variants, compound variants, and so on. And also we have the largest cohort in methylation, which is not only the genome, but also a modulator of the genome, which is called methylation. And that allows us to go a bit more into the modulation of the genome than just sequencing too core.
And Tiago, just a quick comment there as well. And now speaking as a parent of a child with a rare disease, knowing that these programs are out there, it's extremely important. We know that the rare diseases normally take around 6 to 7 years to get a diagnosis, right? And it is based on these programs that actually the patients can have these diagnoses and can act upon them. So as a carer in this case, but also I guess speaking on behalf of those that are dealing with the rare diseases, these programs add lots of value because brings a lot of insightful information that can help a lot, making the life quality a lot better. So this is really just great initiatives.
So, Tiago, from Portuguese to Portuguese, thank you as well for putting your energy here. And in our audience, we have lots of other colleagues that will be interesting as well to join this space. As the director of bioinformatics for the Emirati Genome Program, Tiago, what unique challenges do you face in overseeing such a massive sequencing effort of 1 million genomes? What are the major challenges?
There are, of course, when you're talking about these large programs, there are many large challenges, right? Getting the team together, managing a team, being on top of the literature, getting the cost correct, because just the cost of storing all this information is very, very large. And of course, you need to be very, very mindful of that. And dealing with the finance people that come to you and say, hey, you're spending too much time on this is another challenge. And but of course, the most interesting challenge And I wouldn't say more difficult, because it is what it is how to put this into the clinic practice, right? And as we know, clinical practice goes slow. The whole ecosystem is conservative by nature, and for good reasons, to be honest, because it's hard to do, right? You have so many people coming, you don't have a lot of time, you need to give answers very quickly, you need a lot of patients coming to you. So to bring innovation and to change all this setup is hard to do. And of course we are talking about innovation here, so this has not yet been foolproof, right? There is uncertainty. There is statistics in the way that very, very, very often genomics is not 100%, and the medical profession is not very used to that type of uncertainty, I'd say. So I think those are the challenges. Of course, there is technical challenges there. How do you place this into the doctor's space, right? Is it through an app? Is it through a warning sign? How do you incorporate this into that space is challenging. And of course you have a lot of regulatory and ethical and lawful challenge, which comes to the fore as well, and how the legislatures that very often have no clue of what this technology can go to deal with that is also a major challenge. Lots of challenges, but exciting, of course, to bring this technology forward.
It must be really exciting. With such a transformative program. You've mentioned the people side of things, the mindsets, the technology, and the change management due to the innovation.
Interrompemos aqui esta excelente conversa porque estamos muito entusiasmados em anunciar a primeira edição do Bootcamp Cruzamento. Prepara-te para mergulhar no cruzamento entre a saúde e a tecnologia, com a possibilidade única de aprender com líderes portugueses destas áreas. Além disso, vais ainda ter a oportunidade de assistir ao primeiro live de um episódio deste podcast. Para mais informação, visita-nos nas nossas redes sociais. E por agora, de volta à entrevista.
Talking about innovation, and if we want to kind of then link this back to drugs discovery, could you provide some examples of how genomic data is instrumental in drug discovery efforts, Tiago? And how does this data contribute to the development of new treatments and therapies?
Yeah, I think going to the simplified pipeline of drug discovery, that of course is way more complex than that. But you have kind of the pre-discovery, then you have the discovery, then you have the preclinical, and they have the clinical, right? And of course, each one of these steps moves into a network of complex relationships, but in a linear, it's pre-discovery, discovery, preclinical, and clinical, right? And this space that we are talking about is about preclinical, right? How do you find a mutation, a patient that then moves the field forward, right? And for instance, BRCA1, which is one of the most important genes in cancer, was found through what is a genomic sequencing, although it was not genomic sequencing at the time, but it's kind of the same, where a family of Irish-American women had a very clearly, there was something happening there, and scientists could find BRCA1 gene through that, right? CFTR the same. And so rare diseases that cure blindness in kids the same. So you find, for instance, on the rare disease, you identify that gene, from that gene you can insert an RNA interference, you can insert CRISPR. Now this is the new technology that's already in the market, but it starts with knowing the mutations, it's starting knowing a diagnostic, it's starting understand the family, and all these programs are in that pre-discovery. So let you know way more about the biology, let you know way more about the biomedical situation of individuals, and from there you go to the next step of the discovery, where we try to find something, be it an enzyme substituting the enzyme. If it is something that's interfering in some other gene, and you have a drug, small molecule that blocks that gene, right? So then after what's happening, it's much easier to find treatment. And of course, diagnostics is straight out, right? So if you know the mutations, you can do diagnostics, but the treatment goes from understanding, it's a way into the biology, and from there, it's much easier for biomedical industry and scientists overall to find cures for that disease.
And hearing you, Tiago, for me, all those things are really inspiring. As a biotech engineer, I'm really always fascinated to hear those elements. And we have some, again, for individuals aspiring to enter the field of bioinformatics and genomics. What essential skills and experiences would you recommend they to cultivate to succeed in this rapidly evolving domain? What are your suggestions to our audience, Tiago?
It's a hard one because these things keep changing, right? So sometimes predicting a path is a hard one, right? I mean, I am a biologist, right? I as a kid, I was like to program, right? I had a computer, I programmed. My friends were more into games, and I was trying to program little clocks that would change colors. But then I was a biologist, and then, is it chance? I don't know. I ended up doing a PhD that veered into knowing, like I was doing oxynol guidance in Drosophila, pure genetics, and then microarrays came at the time. I was in Berkeley at the time in the US, and so microarrays came at the time, and so I had to analyze the data, and I started programming because of that. But I'm not a computer scientist by training, right? So, and now I'm a bioinformatician. So it's hard to predict where these things are really going to go. For instance, now there's this idea that you don't need to program anymore, that ChatGPT and more specialized AI algorithms you won't need to be a profound coder. You just need to ask the questions to come up with strings of code that then you can use to do algorithms and that type of thing. So I think being difficult to say you need to learn this, I can talk about the general, which is of course curiosity, reading a lot of papers and a lot of understanding what's around you, being ready to experiment, being ready to push the boundaries, being ready, of course, to have a network of people that do different things. Interdisciplinarity is going to be very, very relevant. And, of course, you need to be very good at what you're doing. I'd say that is something that you really need to push yourself and push your own boundaries, maybe. But I know I'm being very generalist, so I don't think this is... anyone can take great advice from this.
To be fair, I really think it's spot on, and I'm giving as well here my opinion. I do see your path. We admire your path. That's why you are being as well interviewed here, and the curiosity word really stands out. So that's really, really, really good answer. I have to say, Tiago, appreciate it.
So let's look into the future now, Tiago. And looking ahead, you've talked about some technology, CRISPR, amongst others. How do you see the future? So what aspects of the future of the genomics and personalized medicine excite you the most? And what are the elements that you anticipate will have the greatest impact on healthcare outcomes globally?
Mm-hmm. I mean, I think that it's going to be massive. And I think also that we currently will not accept the choices that people in 30 or 40 years are going to make. So that's kind of my prediction. I think we would look in horror in what people are going to accept as standard in 40 years. And I think it's going to be a lot of genetic engineering, a lot of trait selection, a lot of, I don't think that anyone is going to have babies without making sure that the genome is perfectly functioning, whatever that means. That's the future, right? The more immediate future, well, we don't have to deal with all of that yet, but it's going to come soon. But more immediate future is I think that the genomics and this all information, because genomics, you have gene omics, but you have protein omics, you have all these other omics, which is looking into all these biological molecules and trying to understand the cellular state. They are going to come completely embedded into clinical care. So every medical act would be, would start from all this information, and I think that's going to be so it's going to evolve a lot, and we will push very hard into clinical care through that. There is now, of course, this whole story developing on longevity and how you can alter your genes to live longer and live healthier for longer. That might be as well. I'm not so sure how that's going to develop, because a lot of it, to be honest, is a bit less based in science and more based on securing capital to get startups going. But I think there will be something that will be relevant on that space as well, on longevity and how you deal better with this. So that's what I anticipate, yeah.
And maybe before giving it back to Andre, the omics, and we have the photomics as well coming up. So using the Nobel Prize recently, and just a small reference to a startup, as you alluded to, Tiago, a company I love in Portugal that is doing quite a work there. Andre, would you like to ask the next one?
Yeah, and now I'm going to put my patient advocate hat on, Tiago. And as said, I mean, I went through the journey of genome sequencing myself and my family, and I don't have the background of biomedics or the engineering that you are referring to. So to me, it was quite a difficult and complex topic to understand what a chromosome was, what a gene is, and so these were kind of a bit of alien topics. And I must confess that still today, I keep learning a lot about these topics. So it's not an easy topic to be understood by society in general that doesn't have that background. So with that said, how can patient advocacy, how can we bring society be, let's say, enhanced through the integration of genome sequencing in healthcare, so they can participate more in the decision-making process and be at the center of the conversation?
Yeah, first I'd like to say how much I admire your strength and your resilience. I mean, I think the position of parents with rare disease kids that need to take care of them and the love that they need to shed on these kids is absolutely extraordinary. And I admire how you have put all those challenges into a great positive aspect, and thank you for that, because it's actually very important to have people like you pushing that forward. On the patient advocacy issue, I mean, I think that genomics is brought a lot of that technology, funny enough, into the space, right? And even companies, the direct to consumer, Ancestry, Nebula, 23andMe, and so on, actually were a very interesting experiment, how people could get that information and how that could be pushed into the public, right? And also how that information is being so controlled by governments and so on. I mean, I remember in Portugal, there was a certain point where there was a new law, and they asked for people to comment on the law. And I read the law, and I sent them a question saying, I cannot analyze my genome and read my information, because you're saying in the law that this can only go to a medical doctor. So I will have to get my sequence done, analyze my genome, I don't know, close my eyes, not read into what I was reading, send it to a doctor, and then the doctor would have to read it to me. Of course, this is an extreme example, because not everyone would have the bioinformatic capabilities. But it just goes back to say that the idea that you have access to your own genome and to your information outside strictly of a very regulated form is very hard for any legislation in the world, right? And I think it's also wrong because, of course, in a way, patients, it's my own DNA, it's my own disease, right? I should have access to that. I think that has mellowed a bit in the last few years because transparency and access to information has become a bit more open. But just to let you know that I think that genome sequence has helped patients to get a bit more control of what they are doing, and also to push everyone, scientists, genomic scientists, medical doctors, and whatever, to go further, right? Because I think that even the possibilities right now about what CRISPRs and, kids that were going blind, and now there is, cures for disease that just 3 years ago were inevitably going to blindness, for instance. Of course, you have the other side of the coin. Those drugs cost $1 million each, right? I mean, and you can imagine that it's very hard for anyone to come up with $1 million. And then parents are kind of left with, it's just because I don't have enough money that my kid is not going to see. And that, of course, is bringing a lot of new challenges into this conversation. But I think that patient advocacy, as in overall, has been strengthened and emboldened by genome sequencing and access to information.
Tiago, we are coming to an end. We would like to thank you very much for your time and availability and for all these sharings and insights. So, Tiago, thank you very much for making the time today with us. Today, we talked with Tiago about gene sequencing, translating genomic data into medical insights, rare diseases, and the role that UAE is playing with the Emirati Genome Program. Tiago, maybe the very last one, because we want to make sure that people can reach out to you if they like. In case our audience wants to know more about you, where can they find you online?
Oh, okay. So I am not a very I'll say this, very public presence online. I have a LinkedIn page, and I think Tiago Magalhães would be I think people type there, they will find me, and I think that's it really. That's all my public presence. But I will be very welcome, and through questions. I get frequently questions from patients or from parents that find my email online and so on, and I'm always very happy to provide whatever I can. Very often, of course, is through medical, because we are more in the research part than medical, so it's less patient facing. But of course, I'm always very, very happy to provide any information I have. Yeah.
Yeah, thank you very much as well to bring all these complex topics and translate them into such simple language that people can actually understand. And thank you as well for all the work that you are doing on such important program. For those that are listening and hearing us and watching us on YouTube, also thank you very much for staying in with us. Don't forget to hit the like button and subscribe to our podcast for the latest episodes and news. And likewise, connect with us on LinkedIn, Instagram, and any of your favorite podcast app. Check out the show notes for more details, and we'll see you soon on another Cruzamento Podcast show. Thank you.
Thank you. Bye, Tiago.